Thanks very much, Jeremy. Thanks for the invitation to come and speak in this forum. I'm not sure if you can still see the slide that I was projecting.
Yeah, we can. Yeah.
Okay, that's great. Okay, so I feel as though I've got a very hefty dose of imposter syndrome giving the talk on poliomyelitis. As you may be aware, many of you, my main focus of interest and some degree of proficiency is pediatric pneumonia, which is the topic I based my PhD on. But obviously in the pediatric infectious diseases space, we often encounter interesting and challenging cases, and I thought that it would be a good opportunity to share with you a particular case that we treated in 2018 and 2019. So apology to many of those who may have heard this talk before, but it's been updated with regards to more recent developments in the field of poliomyelitis. And I'm just hoping that none of the gurus, such as Melinda Suchard from the NICD, is listening on this particular talk. But if she is, I would encourage her to interject and correct me where I'm running afoul of the current developments. Okay, so just reviewing some of the headline news over the past week. I was actually quite happy in retrospect that I'd been offered the chance to talk about polio because of the tabloid news which had hit the United Kingdom towards the middle of last week and reaching across to the other side of the Atlantic, talking about the polio isolates that had been isolated from sewage works in London, and that the health authorities in the United Kingdom were really quite concerned about this new development and encouraging the populace in the United Kingdom to get up to date with their polio vaccinations. As we're all aware, being actively involved in the infectious diseases space and always trying to outsmart the microorganisms, we always find that we're running a continuous hamster wheel of trying to keep up to date and abreast of the new outbreaks as they occur and the potential threats for outbreaks, even in geographic locales where polio wasn't considered to be of tremendous importance up till even last week. It came with a great degree of applause and accolation during the 2020 on the 25th of August 2020, when the World Health Organization declared the African region as being free of wild poliovirus. And this was decreed to be a milestone in the global fight against this devastating illness. And what was quite interesting was exactly a year later, Melinda Suchard and her colleague Wayne Howard, working quite intensively in the field of poliomyelitis at the National Institute for Communicable Diseases, wrote this cautionary note about the year since Africa declared polio free, but the threat remains. And once again, we cannot ever rest on our laurels in the field of infectious diseases. If tremendous strides have been made, we need to make sure that we implement and maintain the checks and balances which have come to the era of curtailing the spread of wild-type virus or a pathogen. We must never become too self-inflated with pride at such a juncture, and we must always introspect and look at how we can improve surveillance networks and reporting to ensure that we don't have further outbreaks or cases of wild poliovirus on our continent. And once again, in early 2022, you'll see important news coming through the World Health Organization. on the 3rd of March, and then again just last week on the 23rd of June, that wild poliovirus type 1 has been isolated from patients being treated in Malawi and in Mozambique respectively, and that this particular strain was closely related to wild poliovirus type 1, which was circulating in Pakistan. You can see the map here of southern Central Africa, where as a response to the declaration of these cases of wild type poliovirus in Malawi and Mozambique, that Tanzania, Zambia, and Zimbabwe have all implemented preparedness measures to try and increase surveillance activities and optimize immunization efforts to try and limit the spread or the potential spread of wild polio virus again on the African continent. So that's just an introduction to the problem of poliomyelitis. And what I'd like to do now is just to introduce you to a case report that we had the privilege of managing at Chris Hani Baragwanath Academic Hospital way back in October 2018. This was a 9-month-old little boy, and he presented very acutely with inability to move his left leg, and this was of a day's duration. The preceding week had been notable for the presence of a febrile illness, and this preceded his acute presentation of the paralysis of his left leg. Other important history was that he'd had a non-specific cough for 4 months, but no tuberculosis contacts and no constitutional symptoms indicative of active tuberculosis. And just another complaint, according to the mother, was that the child had been constipated over the preceding couple of days. He was an HIV unexposed little boy with an uneventful birth history. He'd been born in a private hospital by cesarean section at 41 weeks of gestation. Unfortunately, his mother was unable to establish breastfeeding with her baby, and as a result of this, he was exclusively formula fed from birth through until 6 months of age, after which time period mixed feeding was established. And his immunizations were up to date. And you can see a copy of his Road to Health card here. He received a dose of oral polio vaccine and BCG at birth, as indicated at the top right-hand aspect of this image. Other important preceding medical history was that he had been admitted at 6 months of age to a private hospital with bronchopneumonia, which required ventilation for 5 days. He had an extended stay in that hospital during the course of that admission with 3 weeks hospitalization. And investigations during that hospitalization revealed that he was HIV negative. He presented with a very severe hypoxic pneumonia. So they were considering pneumocystis jirovecii pneumonia as well. And the PCR for that on his tracheal aspirate was negative. And they also looked for viruses on a multiplex PCR panel. And what was important from this was that his multiplex PCR came back positive for enterovirus and human bocavirus. And the virologist in private, whom I discussed this result with subsequently, mentioned that the enterovirus component detects poliovirus, Coxsackie A, Coxsackie B, and echoviruses. So on his admission to Chris Hani Baragwanath Hospital with his 1-day history of acute flaccid paralysis, we were very concerned about this clinical presentation. According to protocol, he was notified as an acute flaccid paralysis case. He was treated with intravenous immunoglobulin as per protocol, and stool was sent on 3 consecutive days for poliovirus isolation. Unfortunately, he exhibited minimal recovery of his left lower limb function, but was discharged home after 1 week with a to come back date in 1 month for the pediatric neurologist to review him. It didn't take long for the National Institute of Communicable Diseases to report to us that his stool, which had been submitted as part of the investigation for his acute flaccid paralysis episode, had cultured a suspected poliovirus. And this was typed to be a Sabin-like poliovirus 3. And on sequencing, the VP1 region exhibited a 14 nucleotide change from the Sabin 3 poliovirus strain, which indicated was 98.3% similar to the vaccine strain. And the overall conclusion on that sequence was that it was a vaccine-derived poliovirus type 3. Just showing here in the image, so that a schematic representation of what the poliovirus would look like. You can see the structural proteins, the VP1, VP2, and VP3 proteins, which are encoded for in the VP1 region of the genome, which was sequenced in the stool isolates. The other cautionary note, which had been mentioned by the NICD when they looked at the stool isolate, was that we really should consider a primary immunodeficiency in this child. And the child was called back for us to investigate him accordingly. And what we tend to do when investigating in a primary immunodeficiency case is to submit peripheral blood in EDTA, a specimen, for flow cytometry to look at the lymphocyte subsets. And we were very fortunate to have some really experienced clinical hematologists reviewing this particular flow cytometry result. And I want to just try and open up to the audience and those of you who may not have encountered this case before to potentially comment on this flow cytometry result. Do any of the...
Yeah, perhaps one of the fellows wants to stick up their hand. Otherwise I will doom pick someone.
Don't be scared, we're all learning together.
Lyle, I'm picking you. I'm asking you to unmute. There we are. What do you think?
I'll have a look. So it's Lyle here. What the most obvious difference is the lack of total CD3 cells that we made up of your CD4s and your CD8s. And particularly the CD4 component of your total CD3s is low. So CD4 cell is what half of what you'd generally expect. Otherwise, it seems to be a higher percentage of CD8. Maybe it's just as a result of lower CD4 component.
Okay, that's good. So you've picked up some perturbation potentially in the CD4 and CD8 populations. So not ideally within the reference range for the CD4s, and leading to okay, so that's something to note. The other thing I think to look at, and specifically with a potential vaccine-derived poliovirus, is to look at the complement of the blood which supplies humoral immunity or affords humoral immunity. So which aspect of the population of white cells will do that?
We'll be worried about your B cells, so your CD19 positive.
Right.
And then obviously your antigen presenting cells, but there doesn't seem to be, I mean, this is mostly looking at, yeah, your lymphocytes. Mm-hmm. So we're not really seeing your innate cells.
Okay. All right. So what is quite striking here is the normal population of CD19 cells, so the B cells. I don't know if you can see my cursor here on the screen.
Yeah, we can.
Yeah, we can.
Okay. All right. Okay. So anyone else want to hazard a comment regarding something which looks particularly unusual? Okay, I certainly didn't pick this up, and thankfully Debbie Glencross did, but the problem here was the HLA-DR complement of 0%. Don't ask me offhand what the normal range is, but this is very abnormal, having an HLA-DL, the DR the percentage of 0. So as soon as she saw that Prof Glencross did some recollecting of her previous reading around primary immunodeficiency disorders, and immediately arose at the potential clinching diagnosis, but we'll get to that a little bit later on. Okay, so the T helper cells slightly decreased population, and certainly as has been pointed out, a reduced population of CD4 cells. But what was highlighted for us by Prof Glencross was the lack of HLA-DR on the flow cytometry. Okay, when you're concerned about a primary immunodeficiency, not only would you look at the cellular component, we look at the function, or a cursory look at the function. And does anyone want to comment on the immunoglobulins here? This is a slightly easier question.
Immunology is intimidating people.
Please don't be. We're all learning together. I got this. I probably would have got this wrong if I hadn't, if I didn't, care for the patient firsthand.
So why don't people put in the chat then what you think is the abnormalities here, or I guess the interpretation more than the abnormalities. Let's give people another 10 seconds or so to type.
Jeremy, I can't see the chat, so you'll need to fill us in on
no, there's not much options going on. I think people are worried. So I mean, you've got a mildly high IgG. If you in terms of absolute, but probably not dramatically. I'm going to stick my neck out and say not huge important, because high is very mildly high. The IgA is on the low side, technically with the normal, but on the low side. The IgM is helpfully they go less than 0.25, but a normal range that starts at 0.15. So I'm not sure what to make of that, but potentially quite low. And IgE also low. So you seem to have enough IgG and potentially not a lot of the other ones. I don't know. Yeah.
Okay. And then the question is, the obvious question is to try and contextualize the blood draw in terms of previous therapy. So this child was called back about 2 weeks later, and we'd already given him Polygam.
Yeah, so that's a good point. So that should explain potentially IgG. So he may not have much in the way of antibodies at all, I guess, then intrinsically, but you'd have to wait and see, I guess.
Yeah, so I think that's one pointer that you need to just take into consideration. And when we did his nadir immunoglobulins, unfortunately, I didn't include it in this presentation. All of his immunoglobulin fractions were in his boots. He wasn't able to produce his own antibodies. Okay, so all of these findings, so the HLA-DR fraction of 0% and the inability to produce antibody, highlighted for Prof Glencross and also for Jenny Vaughan, that this child probably had a very rare primary immunodeficiency called Bear lymphocyte syndrome. And quite a lot of prior work has been done looking at founder effect mutations in North African lineages that had relocated to France regarding this particular condition. So a very nice article from Wahlberger and colleagues was looking at the impact of mutations in one of either the HMC2TA, RFXANK, RFX5, or RFXAP genes, and how that would impact on your cell surface expression of HL, DR, DQ, and DP molecules. And there's quite an extensive literature looking at this, that there are a number of mutations that affect either one of these genes, and that can impact quite substantially on your expression of these pivotal immune regulatory molecules. A very nice resource for those of you who are keen to explore the immunology and potential primary immunodeficiency syndromes is an article published by Rock, Rates, and Neff in 2016, where they illustrate the immunological perturbations using cartoons. So this is one of the figures from that paper. And this is looking at the normal functioning of your MHC2 complex. So you'll get an invading pathogen. So it's either a virus or a bacterium or a fungus, which is endocytosed by an immune regulatory cell that would be degraded by proteases into its constituent antigens. And the antigens would be presented to your maturing HLA MHC class 2 protein chain. So at the bottom left there, you've got an invariant chain, which is processed in the endoplasmic reticulum. Proteases digest the invariant chain to allow for, in the maturing vesicles within the endoplasmic reticulum, to allow for the antigen to be annealed to that maturing protein. And once the protein and antigen complex has been matured, it's presented onto the surface of the cell so that the CD4 cells can be educated regarding the presence of this antigen, and that in turn will elaborate immune education of your humoral immune system so that you'll be able to produce antibody against that invading pathogen. So we looked a little bit closer at this child's family tree. Bare lymphocytes syndrome is, according to the authors that have investigated this particular abnormality or quite extensively, it's an autosomal recessive condition, and we just took an extensive history in terms of any possible consanguinity between the family members. And there were actually just two very distinct families, which are called family A for the maternal side and for the father side, family B. And no history really of any consanguinity at all. Our little patient's mother was 40 years of age at conception. Father was 43 years, and our little patient was presenting to us at 9 or 10 months of age with this acute flaccid paralysis. Okay, so that's the first part of the case report. I'd like to just move on to some of the nitty-gritty regarding poliomyelitis. This is just a timeline which is derived from this really lovely review article by Rakanielo, who really looked at the past century in terms of development of poliomyelitis and public health responses to try and combat this terrible disease. We are aware that the first records of poliomyelitis actually extend far back into the prehistoric, to the time of the ancient Egyptians, and that these are quite well documented in hieroglyphics. Polio really came to the fore towards the end of the 19th century. It was well known that in the Asian continent, there were interspersed outbreaks of a paralyzing diarrheal disease, which affected the population intermittently. This unknown and much feared infectious disease was imported into Northern Europe and caused extensive outbreaks of paralysis in young individuals less than 15 years of age, and particularly those between 5 and 15 years of age, living in Scandinavia, Northern Europe, the United States of America, and in Australasia throughout the first half of the 20th century, leading to an accumulating number of cases of acute flaccid paralysis, and occasionally involving the respiratory system, necessitating the development of an iron lung apparatus, which was used to try and stave patients over their respiratory paralysis until such time as they recovered from their illness. Unfortunately, a fair number of those patients never recovered from their respiratory paralysis and actually succumbed to hypostatic pneumonias. And it was through 1908 when the poliovirus was first identified, and the first half of the 20th century, that the virus started to be characterized. Intensive vaccine research was underway from the mid-1940s through to the mid-1960s. And two key role players in the endeavor to try and identify a vaccine were Jonas Salk and Albert Sabin. And they introduced an inactivated poliovirus vaccine. That's the Salk vaccine, and Sabin's live attenuated vaccine, which have really been pivotal in terms of dealing with the burden of disease and curtailing the global epidemic of poliovirus, which was very pronounced up till around the mid-1960s. In 1974, the WHO introduced the expanded program on immunization, which included the oral poliovirus vaccine as a backbone vaccine. And the era of molecular virology really came to the fore from the 1980s onwards, during which time there's been further characterization of the virus, identification of the cell receptor through which the virus enters neurological cells, and a really concerted effort to try and, through optimization of immunization policies, to try and eradicate this particular pathogen from the globe. The Polio Global Eradication Initiative was formed in 1988, and we'll speak a little bit more about that later on. So pictures such as this were not uncommon during the course of the 1950s, and this is just a very important a memory of the devastation that this particular virus caused. And I'll just read you the little quotation around this particular photograph. It says, "Stricken with polio, Flossie Rogers is managed has managed a smile as she looked at the world through the mirror of her iron lung in June 1957. So thankful for the care she got from nurses at County Hospital in Mobile, Alabama, that Flossie vowed to devote her life to helping the sick if she ever got back on her feet. Helped with funds from the March of Dimes, Flossie did recover and today is working to become a nurse at the same hospital. So although patients were incredibly severely incapacitated through the paralysis, a fair number of them did survive through really optimized nursing care. And it's an amazing achievement that this particular individual was able to survive in the 1950s. So looking at poliovirus, it's classed within the Bucornaviridae family. It's a human enterovirus C cluster of the Bucornaviridae. It consists of 7,400 nucleotides. It's a single-stranded RNA genome with 3 regions, a 5' NTR with 742 nucleotides, an open reading frame, and then a 3' non-translatable region, which is followed by a poly A tract. Just looking specifically at the genome in schematic form here, you can see the 5' end and the 3' end with the poly A tail. These are the structural proteins and non-structural proteins which are encoded by the genome. And then looking specifically within the 5' non-translated region, there are these loops, and loop 5 and 6 of the genome. You can see that we've got at position 480, 481, and 472, the mutations that confer attenuation in the CNS. So these are the Sabin strain 1, 2, and 3 polioviruses. And through incorporation of these particular mutations, it has taken away the neurovirulence from the wild type poliovirus. And that is what is able to confer immune protection against circulating wild type poliovirus without causing neurovirulence in vaccine recipients. How long is oral polio vaccine or the Sabin vaccine excreted after oral administration? And this was a question that was answered by Martinez and colleagues in 2004, where they looked at 28 healthy 1-year-old Californian children who had completed a full course of injectable polio vaccine, and they were administered trivalent oral polio vaccine. Stool samples were collected at baseline daily for the first week and weekly from weeks 2 through 6. And you can see that the excretion of stool of the poliovirus up to 40 days post-immunization, 20% of the stool samples was positive for the vaccine strain of the virus. But what was of much concern here was that it didn't take long for the vaccine strain to revert to neurovirulence at the position 472. So that's 472 in the genome. So it only took about 5 days or a week before a large proportion of the oral polio strains had reverted to neurovirulence in the stool specimens. Dinsha Tebbins and colleagues are very prolific authors regarding risk analysis in the poliovirus endgame and how to plan towards curtailing onward transmission of potentially mutant orally administered attenuated poliovirus in the Sabin vaccine. And what they did was to look at the literature regarding stool secretion times of the orally administered live attenuated poliovirus vaccines. And you can see that there was quite a remarkable variability in duration of shedding in the different studies that had been conducted. And in patients with immunocompromising conditions, up to a 77-day recorded duration of fecal shedding of the oral poliovirus vaccine. So I'm just going to turn off my monitor and turn it back on again. So that is looking at viral shedding after administration of the oral polio vaccine. There's relatively scanty work done on seroprevalence of poliovirus. And this is a study that was done in the United Kingdom in 1982, looking at archive specimens that had been collected in 1978 across a cross-sectional spectrum of nearly 1,000 UK residents ranging in age between 9 months of age and 80 years. And what they found was that there was remarkable consistency in the seroprevalence rates against at least one type of poliovirus vaccine, poliovirus strain, and about 66% seroprevalence against all three types. And obviously looking at the time trajectory, that most of the patients that had been that were aged 45 years or older during the course of the survey had likely achieved their seroprevalence through wild-type infection. So let's look now at the World Health Organization Eradication Program. This was started in 1988. It aims to eliminate poliovirus, and that is to try and achieve no cases of wild poliovirus transmission from a region in the world, and eventually to eradicate poliovirus. So that's to achieve no cases of wild poliovirus disease in the world, as well eventually to curtail the circulation of vaccine-derived poliovirus strains. Oral poliovirus vaccine is no longer, this is incorrect. It is still administered as part of our EPI as a birth dose, but it is no longer given as at the 6 and 14 week, 6, 10 and 14 week junctures as had initially been devised. It's a live attenuated vaccine providing excellent GIT immunity, induces bloodstream immunity, and what is very important is that it prevents transmission of wild polio from person to person. In April 2016, trivalent oral poliovirus vaccine was withdrawn and replaced with bivalent oral poliovirus vaccine, and reasons for this will be explained in the following slides. Intramuscular poliovirus vaccine or injectable poliovirus vaccine is administered at 6, 10, and 14 weeks in our EPI. It confers excellent bloodstream immunity, but no GIT immunity, and as such prevents disease through the ability to engender a serum antibody response, but it doesn't prevent transmission of the infection. So wild-type polio, the last wild-type polio case in South Africa was documented in 1989. Vaccine-associated paralytic poliomyelitis, or VAP, are rare adverse events which occur as a result of administration of oral poliovirus vaccine. It is estimated to occur in about 1 in 2.4 million recipients and is not associated with outbreaks. And then vaccine-derived poliovirus, VDPV, may be either immune-related or circulating VDPV. Immune-related VDPV occur in children with primary immunodeficiency disorders, such as occurred in our case, which I presented. And circulating vaccine-derived poliovirus occurs in areas with low vaccine coverage, and we'll look at this a little bit in a little bit more detail in the next slide. Obviously, wild-type poliovirus outbreaks would require epidemiological investigation, but in our contexts, thankfully, without detected wild-type poliovirus circulation in South Africa, we really investigate the IVTP and CVDP's cases. So this is just looking at the vaccine-derived poliovirus issue and the requirement for high vaccination coverage in order for us to prevent onward transmission of vaccine-derived poliovirus from person to person. So if you have a patient who has an immunodeficiency and whose vaccine-derived poliovirus causes virulence, if you've got a high background herd immunity through a really good vaccination coverage of the community, because the close contacts will have preformed antibodies, they will not be at risk for becoming infected with the vaccine-derived strain, and will and therefore your the outbreak or the potential outbreak of vaccine-derived strain will not be very concerning or very large or widespread. However, in places where there's low vaccination coverage, the herd immunity is insufficient to prevent transmission of the vaccine-derived poliovirus from person to person. And it is in these settings where you can get widespread dissemination of vaccine-derived poliovirus, which in the community would be known as circulating VDPV. The global targets for polio eradication were missed in 2000, 2005, and again in 2018. And the global trends in acute flaccid paralysis due to polio are currently very low. 3,500 cases were reported annually in the 1980s, but in 2019 to 2022, only around 324 cases have been reported. And you can see here mostly in Afghanistan, Pakistan, the one case in Malawi, and as you're aware, the one case in Mozambique from June this year. So this is from an CDC publication, so slightly out of date. The Polio Eradication Initiative publishes the areas in the globe where polio is still of concern and is causing outbreaks. And you can see that wild poliovirus 1 cases are currently being detected in Pakistan, Mozambique, Afghanistan, and Malawi. Circulating vaccine-derived poliovirus 1 has been identified in Madagascar, and the overwhelming majority of circulating vaccine-derived poliovirus 2, vaccine-derived poliovirus, are related to vaccine-derived polio, Sabin strain 2, which is still widely circulating despite withdrawal of the trivalent oral poliovirus vaccine in 2016. And it was for this reason that the bivalent oral poliovirus vaccine now only contains serotypes 1 and 3, so that we can curtail the onward transmission of serotype 2. Because serotype 2 is such a concern, a new development has come underway within the past 5 or so years, whereby a novel oral poliovirus 2 vaccine has been produced and implemented in phase 1 and 2 trials in 2017 through 2019. Since then, 11 million doses of novel oral poliovirus 2 vaccine has been administered in March through October 2021. And as an initiative to try and characterize the potential for mutation in these isolates. 128 stool specimens were collected through AFP surveillance, and no reversion to neurovirulence has been detected. Remember that for Sabin type 2 poliovirus, the neurovirulent mutation is at the nucleotide position 481, and here the nucleotides that positions for any mutation that was discovered do not involve that pivotal region of 481 for serotype 2. And this is just a recently published article from the CDC. So the novel oral poliovirus 2 vaccine appears to be safe in terms of its efficacy and doesn't pose the similar degree of concern regarding reversion to neurotoxicity of the previously utilized oral poliovirus 2 strain. With regards to immune-associated vaccine-derived poliovirus, Macklin and colleagues are very vocal and prolific in terms of their publication around this particular issue. And this is just from 2 publications from 2017 and 2020 regarding their review or systematic review of the literature, looking at the number of cases of immune-associated vaccine-derived poliovirus, which you can see really became prevalent in our understanding of primary immunodeficiencies, with a gradual escalation in the number of cases detected from 2000 onwards. Of note again is that there tends to be higher stool shedding of poliovirus from primary immunodeficiency patients in high income settings, and patients with common variable immunodeficiency tend to have longer stool shedding than those with SCID or other primary immunodeficiencies. This paper was from 2017, which highlighted the fact that there had been a case with Bruton's agammaglobulinemia, which was identified, who developed immune-associated vaccine-derived poliovirus in Cape Town, and an updated article from 2020 looking at the caseload associated with immune-associated vaccine-derived poliovirus from 2020 to 2019. So the wild poliovirus 2 and 3 were declared eradicated in 2016 and 2019 respectively. Southeast Asian region was declared polio-free in 2014, and the African region, as previously mentioned, declared polio-free in 2020. And has been highlighted by the program coordinators, the final steps towards eradication of this terrible pathogen are proving to be the most difficult. And in response to this, the polio eradication strategy 2022 to 2026 has been devised. It consists of 2 primary goals: to permanently interrupt all poliovirus transmission in endemic countries, and to stop circulating vaccine-derived poliovirus transmission and prevent outbreaks in non-endemic countries. And it's got these timelines for implementation and review of the program. And what is quite interesting in goal 2, as mentioned before, was the use of a novel oral poliovirus 2 vaccine in order to try and confer optimized GIT protection against serotype 2 without incurring the risk of neurotoxicity. So the eradication program looks like this in terms of their overall framework: permanently interrupting poliovirus transmission and stopping circulating vaccine-derived poliovirus transmission. Goal 1 would need to include political advocacy, community engagement, campaigns to recruit and train and appropriately support motivated workforce, to optimize the the efforts to disrupt the transmission of wild-type polio, to integrate all the services, and to strengthen surveillance. Goal 2 is the goal of disrupting circulating vaccine-derived poliovirus, also using the same avenues of intervention, engaging government and political advocacy, strengthening surveillance, deploying novel oral polio 2 vaccine and improving campaign and planning execution of coverage of this vaccine, to promote community engagement and to integrate the activities. It's important to try and pivot away from Sabine oral polio vaccine 2 to novel oral polio vaccine 2, to move to a regional structure of reporting and to use to try and optimize the involvement of both women and men, but to really promote the integrated means of curtailing circulation of vaccine-derived poliovirus. This article was published relatively recently by our colleagues Wayne Howard and Melinda Suchard, working at the NICD, where they reviewed the adequacy of our acute flaccid paralysis program. 2 cases of immunodeficiency associated vaccine-derived poliovirus were detected in 2016 to 2019. The AFP reporting rate was 3.1 per 100,000 persons under 15 years of age, which was within the WHO requirements of 2.0, sorry for the typo there, per 100,000. And the adequacy of stool samples in investigation of the AFP was suboptimal, however, and needs to be optimized. The other new development is that of the potential use of antivirals. Polio antiviral drugs are in development. They control poliovirus outbreaks in the post-eradication era, and there may be an immediate need for use of these antivirals in immune-associated vaccine-derived poliovirus cases, in terms of the personal risk of poliovirus in the patient, and to minimize the risk of delayed polio eradication through ongoing stool transmission from these immunocompromised patients. Polio eradication will not be complete until all poliovirus excretion has ceased. These are 2 candidate molecules, Kappavir and V7404, and they target different aspects of the viral assembly pathway, Kappavir specifically being a capsid inhibitor. And this is the position on the VP1 capsid protein where Kappavir binds. There has been successful use of Kappavir in terms of clearing immunodeficiency associated vaccine-derived poliovirus in the patient that I mentioned earlier on that was treated by our colleagues in Cape Town in 2019. And this child had successful clearance of poliovirus excretion from his stool after administration of Kappavir. But back to our case, we were able, through our application to SAPRA and the provincial drug committee, to access Kappavir for our particular patient. And we were able to identify or quantify the mutation rate of the viral isolates in the serial stool samples that we submitted to the NICD. You can see that of the 26 stool samples submitted to the NICD, there was an initial mutation rate of about 2% of the viral genome prior to the use of Kappavir, and this plummeted to less than 1.5% during the phase of administration of the Kappavir. And during the phase of the administration of the Kappavir, we took daily stool samples, and you can see the downward trajectory in the number of mutations that were detected in the stool. But what was of concern through our case was that when we discontinued the Kappavir course, we had a rebound in terms of the mutation rate, up to 3% mutation rate in the stool, which subsequently settled. Unfortunately, in our case, despite the intervention of regular polygam doses, the use of Kappavir, you can see that a very important indicator in pediatrics is the weight trajectory, which we were not able to get on top of. He developed ongoing paralysis and eventual bulbar palsy and was not able to retain feeds. And as a result of this, unfortunately, he had an aspiration pneumonia event and unfortunately succumbed to his illness. Subsequent to his demise, we got the results of the study of his genetic study, and you can see that he had an RFX5 mutation, which was a pathogenic mutation, which was affecting his ability to produce the HLL-DR receptor, which confirmed that he was in fact a case of bare lymphocyte syndrome. Okay, so that's the case. I'm sorry I've gone over time. I thought we'd have time for questions and comments, but I'd just like to acknowledge my wife, children, and family, the Carnegie Corporation of New York, Prof Valapi, and my colleagues at the Department of Peds, Chris Barra, and our little patient and his parents. Thank you.
Great, thank you so much. That's a really, really lovely overview and an incredible case. It's astonishing how you, as you said, you basically found two rare things in the same patient, clearly associated, but really, really astounding case, and you know, really amazingly worked up and presented in such great detail. I've got there's some questions that people have messaged me and sent in the private chat. If you have any other questions, just type them into the chat. One question that got sent to me is why are we using a bivalent vaccine? Couldn't we just use a monovalent? Is it just that it's not much point given that subtype 3 isn't hugely troublesome?
Yeah, so something I didn't include in the talk was the concern regarding waning immunity to OPV2 because of the usage of bivalent OPV. And this has been seen in China in a recent paper to be perpetuating the potential for circulation of vaccine-derived poliovirus 2. And that's why there has been an initiative to adopt the use of the novel oral poliovirus 2 vaccine to try and confer mucosal immunity and prevent onward transmission of circulating vaccine-derived poliovirus 2. So yeah, sort of doing a dance between the potential for circulating vaccine-derived poliovirus and maintaining adequate mucosal immunity to prevent infection if there is onward transmission of vaccine-derived strains.
Yeah, so difficult to time it right, as you said. It's really difficult. And another question that came through was about the stool sample adequacy, because I think it was 53% or around about there that was adequate. In what way is, because adults don't have much to do with this, but in what way is it inadequate? What do you have to do to make it adequate, the stool sample?
Yeah, so there is quite a strict protocol in terms of making sure that the stool reaches the reference laboratory in time and in fit condition. So I think there's a time period, I stand to be corrected on this, between 48 and 72 hours, but the stool needs to reach the referral laboratory on ice and adequately cooled. So that's the one aspect, and the other is the 3 consecutive day stools. Doesn't need to be consecutive, but it can be within 42 to 72 hours, but you need to submit the 3 stools initially. and that's often not adhered to, unfortunately. So it's looking at stool coverage in terms of adequacy of sampling, as well as the condition in which the specimen reaches the referral laboratory. And I remember for our particular patient, I used to drive stools through in cooler boxes to the NICD to make sure that they reached the cooler lab there. They've got a huge cold room in NICD.
Sure. And then just a final question that came through is was about, and I think that I did read something about this a while back, but that there is interest in developing other forms of oral polio vaccine which don't revert to vaccine-derived polio so easily. In other words, presumably of multiple mutation steps away. I don't know, is that something that you've kind of come across, and is that anything on the horizon relatively soon, if so, or do you think we may eradicate it before we ever need it?
Yeah, so I think that the main point regarding that particular issue is the novel oral poliovirus 2 vaccine. So that has been piloted and is due for implementation as part of Goal 2 in the updated 2022 to 2026 strategy. I think that I would encourage you to invite the gurus in the field, such as Melinda, to give you real insight to sort of the machinations of the advances in the program with regards to curtailing the vaccine-derived poliovirus aspects through use of those genetically engineered variants. But the novel oral poliovirus 2 vaccine is the one new kid on the block, which is due for more widespread implementation towards the end game of poliovirus.